So Zhugen, maybe you can now share your screen, and we're going effectively to move to the biosensor approach, and going to have some example on how we could use this sensor device to get real-time monitoring. Over to you guys again.
Okay, thank you very much, Fred. Yeah, I will slightly mention about the chemical sensors, but mostly for biological sensors. I just get.
Yes, you're all good.
Yeah, okay, so I
no,
just.
You're back. You need to be back on diaporama.
Yeah, okay, maybe I first turn off my video and then coming back. Yeah.
Yeah.
Okay.
All good.
All right. Thanks very much again for all the people coming and nice invitation. And today I will just share you with my work on the paper sensors for diagnosis of infectious disease and wastewater-based epidemiology. So I will firstly give you a brief introduction about what we are working on in Cranfield Sensors Lab. I'm really grateful to get this brand new lab in Cranfield University. Although in the history, Biosensors has a good track record and a big impact in Cranfield University. But from the Water Science Institute, we are just set up a new laboratories, which was funded by UCRIC. And our research activity mainly including 3 parts. So our vision is try to better understand the environmental health in order to improve human health. So the technology is covered a few biosensors, including optical sensors and electrochemical sensors. We are mainly look at first is for wastewater-based epidemiology, from chemicals to microbiologicals. Recently associated with SARS-CoV-2 crisis. And second part, we are also interested to develop nano biosensors for diagnosis of water and environmental health, including some microbial contaminations in drinking water. We do have some project which was funded by Royal Academy of Engineering to promote the low-cost and rapid sensors for water quality monitoring, including microbial pathogens and antimicrobial resistant genes and antibiotics. And the third part, we are interested to develop rapid and point-of-care diagnostic sensors for biomedical disease, including infection disease and cancers. First, I'm just want to share you with some, this is outline. I will give you 3 parts. First, introduce some background about biosensors, then give you examples to demonstrate how do we move diagnostics from the lab to the field using our Paper-Origami device. Finally, I will give you some overview about our activities in SARS-CoV-2 testing, which in link with the National Monitoring Program, and Barbara also in there as a partner. Many of you already know some sensors. As you see from the global market, you have see that it has a wide attention in terms of using biosensors, and it's great not only for medical diagnostics, but also for other areas, including environmental science. So it's increasing year by year within the requirements. As you see in we have already get very successful commercialized products. One is the glucometer, another is the paper pregnancy test. So we are hoping that we can get all the chemicals, biologicals, or contaminants can be used as a glucometer, a similar sensor to report the or diagnosis the results very fast and on site. So what exactly is a biosensor from academic terminology? So basically, a biosensor is an analytical device combines of biological component with a physical chemical detector. So in terms of exact application, you can simply categorize with different sensors. For example, if you want to test proteins, you can say it's protein sensor, a DNA sensor, or molecular sensors. So from the exactly the component, we can simply clarify with analyte, which is a target you are going to test, and a bioreceptor, which is the probe, you need to interact with your target. Then the third part is a transducer, which means that we need to translate this bio recognition signal into the readable optical or electrochemical or mechanical sensors. So one of the most important is that the surface chemistry, in order to efficiently immobilize your receptor on your substrate, then get the biological readable data. So in terms of basic fundamental scientific questions related to sensor is the sensitivity and the selectivity, which is as analytical tools. So this is a really very important question before you can establish and using these sensors. However, compared to some classical analytical tools, sensors typically have some advantages, including rapid response time, which is very cheap for the usage and easy to use, and there is a potential for field testing. So that's our people who are working in this area to try to improve these sensors. In particularly for field testing or for being used in in developing countries, there is some also some important factors which we will try to improve, both from the analytical science and engineering aspects. So which as I just slightly mentioned, which should be a SUD principle, which was followed by the WHO guidance. As we already get to know a little bit background of biosensors compared to analytical conventional analytical tools, we have some advantages. In specific case for the detection of pathogens, so there is still some challenges. So typically people are using culture method, which is time consuming and limited in the lab. Alternatively, people can use molecular method. I think more and more people already get to know the polymerase chain reaction because of SARS-CoV-2 crisis. So it's still lab-based method, typically very expensive, especially during pandemic time. There is a problem of shortage of reagents and lab access. Also, the data need to be interpreted by a skilled person. So typically, the turnaround time will be 24 hours or 48 hours. Those both limit the application in biomedical diagnosis and the environmental public health. In this sense, there is a great need to develop a rapid, low-cost, sensitive, and a portable platform to rapidly diagnose these samples in order to get immediate information. So I introduce you with our earlier work in terms of developing Paper-Origami sensors. Initially, we developed these sensors for the diagnosis of infection disease in India, but because India farm suffers problems within infection disease may be rapidly spread in the whole farm, we want to develop a rapid and field-based testing to better understand these infections and limit the spreading. Otherwise, the whole produced milk will become the waste. So we focus on three common pathogens associated with sexual transmitted infection of buffalo and bovine. So then we develop a wax-printed paper analytical device to rapid test three pathogens and read the data using the colorimetric method by color changes. Here, the green spots represents negative and blue positive. Blue represents negative. And I will show you how exactly it works. Basically, it contains three components. First is a paper device, which is fabricated by an office wax printer, which can generate hydrophobic and hydraulic layers. So here, the black layer is hydrophilic, which is wax, and we can manipulate these samples from this paper device by capillary force instead of using typical microfluidic technology using a pump. So we don't need any power, can drive, but we can still drive the samples through different layers in order to get the sample processed. It also contains glass fiber, which can absorb or adsorb the nucleic acid within the electrostatic interaction. So the third part is this plastic cassette, which can accommodate the amplification. Here we're using the lump method, which is loop-mediated isotome amplification. So recently attract increasing attentions. I don't need to go more details about this amplification method. So what we do is that we first get samples licensed and introduced onto this sample device. As we mentioned, because of the electrostatic interaction, the nucleic acid can firstly absorb on this glass fiber, and then we using after washing off the nucleic acid residues, then we can elute those this nucleic acid into this small cassette, and then to distribute into several channels to achieve the multiplex detection. Afterwards, we add the primers and enzymes and stay for half an hour. Then we get the results detected within color changes. So the whole assay from the sample in to data out can be achieved within 1 hour. So here I have a very small video, which quickly to show you how exactly it works. Right. So there's another question, especially in terms of environmental science application. People are more concerned about the quantification. So this paper device can be rapidly test you yes or no within the color change. However, we can still capture the image from the tested data and using software to achieve the semi-quantitative detections. Here is just to show you an example about testing 3 pathogens and controls by extracting data density and then to identify the quantitative results. To give a quick sum up, we actually demonstrated a really low-cost paper origami device. We roughly evaluated this cost to maybe less than £1, and the sensor itself can achieve a rapid responsing and data collection. So the whole assay can be less than 1 hour. It's really user-friendly. So here is a picture. When I went to India to do the field test, after training the local student for a couple of hours, so they can do this experiment independently for this testing of semen samples. So most importantly, it has a significant potential to test those pathogens in various clinical samples or environmental samples in the field. Inspiring with this success of the field testing, we try to extend this application to more other diagnostics. Working with colleagues in Glasgow University, we further develop this technology by changing the readout from the calorimetric detection to the lateral flow device for particularly biomedical diagnostic application. So here I give you a very quick impression of yes or no signals. So we developed this a new paper folding and unfolding device and validated these results in the laboratories and went to Uganda in Africa to test the malaria with a local primary school. So we found that our Paper-Origami device shows even promising sensitivity and specificity compared to the conventional method. To quick sum up, so we basically develop a paper-based platform to test a range of microorganisms, including pathogens and associated with infection disease, and some such as E. coli, Salmonella, Brucellus, different species. So this platform is a molecular-based method which can identify species specific assay, and the most advantage of this method is very rapid, cheap, and can be used in the field. So we actually also be active to use this platform in different species, including water, wastewater, and soil for environmental application. So the next questions, I just want to show you some our efforts on the using sensors for the wastewater-based epidemiology. So as Barbara mentioned, a lot of the chemical monitoring using wastewater-based epidemiology. So we are I'm very interested to further extend this technology combining with sensor in order to achieve the field-based testing. So here I'll just highlight that we actually dating back to 5 years ago, we proposed these ideas and published at ESNT papers to using community sewage sensors for monitoring public health. So here we're including chemicals within different, including illegal drugs and pharmaceuticals, but also some pathogens associated with infection disease. So recently it's been fast emerging as a promising technology to testing sewage in order to mapping hotspots of SARS-CoV-2. So here, just very quickly to show our efforts on the detection of chemicals and drugs. So we are using alternative technology, including electrochemical sensor. Here, just Barbara also mentioned that we 4 years ago we published this paper on detection of cocaine in different sewage. We also found a similar weekend peaks in terms of the illegal drugs, which is in agreement with other European studies. And very recently, we also developed a Raman spectrometry sensor using nanoparticles for signal enhancement. So we're using a portable Raman spectrometry, which we also have in Calendar Lab, and using nanoparticles to enhance the signals to detect methamphetamine in Beijing. And we map the drug consumptions within different areas in Beijing, urban areas and rural areas. So we also have a combined study with in European Centers of Drug and Drug Additives, EMCDDA, and within cost framework to get this funding support. So this is very brief about these sensors for using for the chemicals. But again, as Barbara highlighted, it's very challenging to develop really sensitive and field-based sensors in terms of for align detection. But we are making efforts to improve. So here is some impact about this wastewater-based epidemiology. It's fastly emerging as a global technology, in particularly to test sewage for SARS-CoV-2. Actually, just 2 years ago, it's also being highlighted in Nature News because this method has been used and deployed in China to fight against the illegal drug crimes and help the police to successfully seize some criminals. And also after the SARS-CoV-2 crisis in this April, there is another highlight in terms of testing sewage for the after KWR in Netherlands to test positive in Amsterdam airport. This could attract more and more attention. So technically, as you know, people already think this is a really promising technology. In terms of technical analysis of this sewage or SARS-CoV-2 virus in wastewater, the typical workflow relies on the transcript real-time polymerase chain reaction. So the whole workflow including 4 parts. First is the sample enrichment. As you know, typically people need to take 100 or 500 milli sewage samples. So the first step is to try to enrich those RNA from these sewage samples, and then following up with the ion extract using commercialized kits. Then third step is run the PCR amplification. Finally, we need to interpret these results. So the typical workflow takes 4 to 6 hours, including some complex sample concentration. It may take longer. Although people are working hard to improve this turnaround time, but typically it still takes 24 hours or even longer. So this is just to quickly show you this. I think this is the first peer-reviewed paper which is reported from University of Queensland in Australia in April. The question is, can we integrate all these steps into a single device which can enable the more fast and field-based testing? So by inspired this, we try to look at if we can use our paper-based technology to detect this SARS-CoV-2 in sewage. So we firstly proposed this idea and published this paper in March, which is even I think within that Nature highlights. So then we think about from 2 points to improve this technology. First is to do the rapid testing in terms of analysis of this viral RNA. Secondly is we need to think about how do we enrich these sewage samples, because the sample size will change from a large sample rather into the microliter level samples. So it's also being fetched by the science news, because in Europe, the first report from the Paris, they report the test positive from the sewage. So, and also we, because of these good contributions, we also been invited to attend some forum, SES publication forum, to discuss about these technologies. How can we improve this testing and diagnostic technology for SARS-CoV-2? As we mentioned, for the sensors to testing sewage, there is a big challenge is that the samples size changes. If we test the clinical samples, for example, blood samples, the microliter level is enough. However, if we want to test the wastewater samples, typically we need to collect 100 millisamples or even more. So how do we using paper device to test them after the sample concentration, or can we do the sample concentration without any power, without any centrifuge, or without to get a lab-free facilities? So we developed this syringe-based method, which is to concentrate nucleic acid from the wastewater and using a magnetic beads to purify those nucleic acid and then to make a lump reaction, so which can be achieved within 60 to 65 degrees typically. So we're using a thermos to provide this temperature in order to make this reaction happens, and then using a lateral flow device to test the positive for this for the proof of concept study. So based on this analysis, we don't need any centrifuge, we don't need any power, and centralized laboratories-based facilities. We can still test those nucleic acid from the wastewater. So we are very inspired by this track record, and then try to help to test the rapid test SARS-CoV-2 in wastewater. So this is more work about that method. We prove of concept study a cohort of sewage samples from the local treatment wastewater treatment plant, and also we are able to quantify those nucleic acid loads based on the flow meter. So as you can see that within the flow meter increase, the concentration just decrease. So this is just because the local population is fairly constant. So the shedding of the DNA is not very very much rise. So we are hoping that based on this sample concentration method and Paper-Origami device technology platform, so we want to our ambition is to try to develop a rapid and on-site platform to map these hotspots or to contribute to this wastewater surveillance in the UK. So then, as we working together with the partners from Scotland, England, and Wales, so we are getting funding to support this study and associated with different organizations and water companies. So specifically from the Cranfield contribution, we try to deliver this origami diagnostic method for on-site mapping these hotspots. And we are currently working very hard to improve and get, and we are analyzing samples within our local water companies and as well as local wastewater treatment plants based in Cranfield universities. So hopefully I will be able to report to you more results by sensors. We basically already proof that sensors can be tested different viruses in wastewater samples, and we also test positive using this concentration method and the paper platform. Just a quick sum up. So we basically focused on the development of a range of sensor technology to address the challenges in water, environment, and health issues. So the technology, including, as I showed you just now, recently mainly focused on the Paper-Origami device, which enable the rapid and rapid and field-based testing of different pathogens. And we tested different species for a range of applications, including biomedical diagnostics and environmental health from different species. And we also working hard for the chemical sensor development from the illegal drugs to antibiotics in order to understand those correlations in the environment. So most recently, we contributed for SARS-CoV-2 sensor development in order to mapping those hotspots in sewage to provide early warning sensing systems. Finally, just give you a very quick impression that we recently some contributions about the sensor technology for SARS-CoV-2, both for biomedical diagnostics and for sewage testing. So we recently also proposed integrated biosensor systems within mobile health for wastewater-based epidemiology. So the idea is that we try to collect those signals very fast and then to help the organization to make an immediate decision in order to do the proper measures. And we also summarize the challenges for biosensors and nano-biosensors for the biomedical diagnostics applications, because there is always a significant requirement for mass testing kits with cheap, low cost, with cheap and faster turnaround time. And we also summarize the wastewater-based epidemiology for drugs and to evaluation drugs for the potential and for the wastewater using sensor technology. Finally, I would like to thank my group members, collaborators, and sponsors. And we are just growing a group which just attract several new students. And finally, just give a quick advertise. We are recently doing a special issue, most specifically focus on biosensors and a new analytical method for wastewater-based epidemiology, which is in a good journal in analytical chemistry field, Trends in Analytical Chemistry. And any people who are interested to publish a review paper, so you are very welcome to invite for these contributions. Thanks very much.
Thank you very much, Zhugen. As you can tell, I'm taking over from Fred, who had to leave at about 1 o'clock. Thank you again, Zhugen. Very interesting talk. Wonderful technology there. Let me just bring up my video so people actually know who I look like. Also wonderful talk there, Barbara. Both excellent talks today at this webinar. I have been receiving a few questions in the chat line, and I welcome everyone who has a question to plug in their question on the chat line. There is Zhugen, if I can just pop over to Barbara right now, as there is a question there which was posed and wasn't answered. Let me see. It was I think by me, in fact. Oh yeah. So, Barbara? What's your perspective on using this approach you have with wastewater, looking at wastewater metabolites and so forth? This approach to understand and resolve the COVID pandemic.
So we are at the moment. This is just, so many questions and very few answers at the moment. And if you ask anyone working in this area, I'll provide you the same answer. So at the moment, we are testing for RNA, and as a result, we don't cannot distinguish between SARS-CoV-2 coming from other sources not related directly to human, and there are other ways to test. How many people got symptoms, were affected, by looking at some of the indigenously formed markers. We are, for example, looking at CRP, C-reactive protein, in wastewater. It is a very challenging measurement because it's a large molecule and it's not as stable. Different tools have to be utilized. So there are some challenges that I'm sure they will be overcome, and there is no, at this stage, a solution to basically be able to pinpoint RNA in wastewater directly with human. Now, we're also looking into sequencing. So while I'm a chemist focused on mass spectrometry, we also have microbiologists that do sequencing, and I see the future will lie in combining these two powerful tools together. This is on top of sensing. We know that sensors will catch up. We have great people working in this, such as Zhugen, developing the tools. But before we know what to sense for, we need to understand the system. So the high-resolution mass spectrometry and sequencing is really the way forward in trying to understand what kind of information there really is in wastewater. We are just at the top of the iceberg at the moment.
Yeah. Thanks, Barbara. And there's just one question that came in as you were answering that. I think this one's directed to you. It says, silly question. I just want to say to Joanne, there is no silly questions. But what are the other sources of SARS-CoV-2?
The other sources. Oh, yes. So what we want to do in WB is to be very quantitative about this. So we want to make sure that RNA copies that we see in wastewater, and then they can be normalized and everything, and per head, per capita. So coming directly from us, and ideally linked with us humans, then we can also have exposure from runoff, so various different animals. But one very important aspect, which is really giving us a headache, is that SARS-CoV-2, inactivated SARS-CoV-2, is used as positive control in the labs analyzing for RNA. So we might be hitting some false positives if there is a lab in the area that is using SARS-CoV-2 as positive control. So this is one of the sources we would not like to see in wastewater. But by definition, wastewater, we have just everything that community produced, and we will find a way to take this into account. It's just we need to have some time, I guess.
Yeah, very good. Thank you, Barbara. Over to you, Zhugen. I have a question here from Louise. Are you looking at any environmental monitoring applications in particular? Which contaminants or situations would you look at first? In fact, that was a question I had noted to ask as well, because I'm very interested in the environmental aspect of your technology.
So we basically look at a broad array of the contaminants, which including chemicals and organisms broadly. So specifically, we mostly look at the some pathogens which relates to drinking water contaminations. For example, E. coli, salmonella, or cryptos. So these pathogens classics. And for the chemicals, we have done some work within illegal drugs. And recently we moved on to antibiotics. So using this different sensing technology, including this paper-origami device, but also within electrochemical sensors. So we specifically focus on the development of paper electrodes within different chemical bio modification in order to improve the sensitivity and the specificity. So we do have the portable electropotential start, which can achieve this rapid testing chemicals. We're working from the antibiotics, but we extended to some pesticides and disinfection byproducts. So this is also of interest. And also recently from some plastics using surface enhancement Raman spectrometry, because there is a lack of the technology recently using people use IR or microscope. So we are trying to using this Raman spectrometry to improve these particles, including microplastics. Yeah.
Very good. So yeah, I'm also working on some microplastics projects, looking at pathogens associated with plastics. So we've got some projects starting off there. So it'll be interesting to have a discussion with you about that.
Yeah. That's in the plan.
So the other environmental monitoring aspects or of interest with using your technology could be signatures of microorganisms in the environment that could be what I'm saying is markers of climate change. So that might be one to think about as well for your technology.
Yeah, definitely.
Yeah, I had another question here from Louise, just trying to go through the chronology here on the chat line. The question is, if an industry wanted to demonstrate compliance with particular environmental regulations, could this help them?
You mean using sensors?
Yeah, your sensor, your paper-origami technique. So if they were like, had a regulation to fit compliance, like the detection of a pathogen at a particular quantity or detection or not, to fit that compliance, would that be of use to industry?
I think that's a very good point. I did have some engagement with water industries. They are interested to deliver some online monitoring, including sensors for example, for pathogens monitoring, especially because of the aging, because of the leaking. There is some growing on the pipeline during the drinking water transportations. So if we find that outbreak, they need to pay a lot to kill those, for example, cryptos or some other pathogens. So we try to help them to develop this online method or rapid. This is also one of part of my fellowship grant, associated with Scottish Water. In terms of fully regulated, so I think there still need to a way to go in order to standardize them and recognize that the levels in terms of the performance, sensitivities, and specificities to meet that requirements. But I'm quite confident within this sensor to detect pathogens in particularly, because we have a really promising sensitivities to meet this detection challenges, I think.
Okay, thank you, Zhugen. To Zhugen, I think this question is for you. This one's from Fiona. Do whole cell biosensors have any future for measuring bioavailable pollutants in wastewater?
The whole-based sensor.
Or it might be for Barbara as well.
So I just some thought in terms of the whole cell-based sensors. I think it's it's attracted a lot of attention in this field as well. So especially recently in terms of the emerging synthetic biology technology and some CRISPR, very it's a Nobel Prize level technology. I think wholesale-based technology can still have a lot of breakthroughs or innovations, particularly for the application of the environmental contaminants. But I think sensors always have a big demand, both from the academic or the industry. So if you can have some great ideas into make breakthroughs, I think it definitely can meet different applications. Maybe God can come on the wastewater-based epidemiology application.
Yeah. Barbara, did you have any comment to do with that?
I think I will stick to what Zhugen has just said. We've been looking at sensor development over years, and obviously worked with Zhugen on some of the approaches. I can't wait really to have sensors that can deliver and all the complexities that we are focused on at the moment. So we are not there yet. That's why we use this tools that are been developed years ago, such as mass spectrometry. Would like to move away, and also yes, linking exposure and effects from an environmental point of view. We really have to move away from targets and try to evaluate the sample as a whole, both from what is in there in terms of chemical makeup, but also what this means in terms of biological response. So there is something that we have not really tackled today. So the kind of interface between the two. So some sensing approaches might be able to deliver. So touching upon toxicity dimension and our various different responses that might be of interest to us in evaluating environmental impact.
Yeah. I had a question for you, Barbara, and I apologize if you had mentioned this during the talk and I missed it. Do you, in your sampling program of wastewater, do you specifically target particular times in a 24-hour clock? So do you go for mornings or evenings when people are mostly going to the bathroom, especially in the mornings, or is there a particular timing that's of interest?
So you're right, I have not mentioned this, and I can't believe I haven't mentioned this because it gives us a headache. Yeah, we do 24-hour composite sampling, and this is flow proportional composite sampling. If you don't do your sampling right, you will get results, but they won't be meaningful results. Yeah. So yeah, definitely. And we collect sample every few minutes throughout 24-hour period time. Saying that, not everywhere we have availability of samplers, especially in low resource settings. So this is where passive sampling approaches might become of use. So yes, active sampling, 24-hour composite, flow proportional, this is what we want, but it's not always available, and we have to be very innovative in this respect. And sometimes we simply collect a grab sample and result is more qualitative than quantitative. But yeah, again, an innovation in there is still needed. So how do we sample in a way that is meaningful? And also samplers, as we know, are extremely expensive, and in various locations worldwide we cannot afford them. So innovation here on top of sensing would be very much welcome. Yeah,
can I just add a comment in terms of sampling? Actually, as Barbara mentioned, it's typically when people do the chemicals, we're usually using 24-hour sampling, which means that we can collect all the information. We never miss any samples. However, because of this wastewater-based epidemiology emerging as a tool for monitoring SARS-CoV-2, because of shortage of this supply of the auto sampler, people already try to grab samples to test as quantitative to show the presence of these SARS-CoV-2 particles in the sewage. And I did come across some groups are looking at the different time series of this sampling time, as Tony concerned, for example, in the morning or in the afternoon, or whichever time series which has a good for sampling, because that will definitely facilitate your sampling. Not every group or every people can have access of those auto sampler. I saw there is some it's still on the investigation, but there is some conclusion, partially from American group, I think from Arizona. They are working out some timing is 11 o'clock. It's good for the SARS-CoV-2 detection, but it varies from the treatment plans and the local population and the sharing of this urine or these biomarkers into the sewage. But it's interesting, you know, if we can get rid of those in terms of 24-hour samples for the complementary information, but we can grab a very accurate sampling point and a sampling time. That will be definitely facilitate this testing. Yeah.
Yeah. No, thank you very much, both speakers. So we've actually come to the end of our webinar. So it's been jam-packed with two wonderful talks and lots of questions there. So I'm going to pass it over to Angie or Louise to end the webinar. Thank you to both of you participants, both of you speakers again, and to all participants for attending today. Thank you.